And so when we're deciding whether to make very dilute or very concentrated or just very average run-of-the-mill urine is always dependent on the needs of the body. So the primary means that substances are moved in and out of the nephron loop after that original glomerular filtration is through active transport. So remember back to your sophomore bio days that active transport refers to transport that requires ATP or energy in order to move substances from where they are already concentrated, and we're going to actually increase that level of concentration gradient. So that is why it requires energy, is because it's kind of going against what nature would prefer. Normally, diffusion will go from areas of high concentration to low. Active transport is going to maintain or exaggerate that gradient that has already been produced. So we might have a little bit of urea and we want it to be even higher. We might have only a little bit of water left, but we are going to actually use countercurrent multiplication to make sure every little bit of water does get reabsorbed. So the three specific types of active transport systems found in the nephron loop are first going to be the symporter. So the prefix sym means same. So in a symporter, two substances are being pumped in the same direction. So our example here is sodium ions and glucose. That is a very common pump. Both of these are very important to the functioning of your body, and they are substances we do not want to excrete. We always want to keep them in because they can be reused and recycled. Antiporters are going to have two substances that are going in opposite directions. So the sodium potassium pump is actually a great example of this. And actually the sodium that is pumped out with the sodium potassium pump is actually used to drive the reabsorption of glucose. So a lot of times these systems will work together in order to produce the desired results that we want. But it's anti because they're going opposite directions. One is being reabsorbed and the other is being secreted. And then the end result of this is what we call countercurrent multiplication, where basically two tubes with very different gradients are next to each other. And so what's happening in one tube helps drive or amplify or multiply what's happening in the other tube. This is used with urea and some other, especially sodium ions, to create that really salty medulla that helps us to reabsorb water really, really effectively. So all of these gradients are used to allow us to do passive osmosis in order to reabsorb more water whenever necessary. So here's some diagrams of some of these types of processes here. So this is an antiporter. So we have sodium that's being pumped out. So this would be from the renal tubule back into the bloodstream, and then potassium is going in. That's the same pump that we use in our neurons or our muscle cells to create an electrochemical gradient to send messages. And then that sodium, after it's pumped out, actually can be used to drive the reabsorption of glucose. So these two systems do work hand in hand and are both really, really important for water balance and for making sure that we don't lose materials that are actually really helpful for our bodies. So diffusion, facilitated diffusion, and osmosis, of course, are all passively going to play a role in managing what substances are added and which ones are removed from the filtrate. And remember that filtrate, whatever remains in it, will be excreted down through the ureters, the bladder, and eventually the urethra. We also have capillaries that are crisscrossing all through this renal tubule that allow tissues to receive nutrients and also to provide those substances that are secreted or reabsorbed. And so all of these tissues and all of the cells that make up these tissues are always going to be receiving oxygen and to help to wash away the water and drain that water back into the general circulation so that those gradients don't get washed out. So here you can see the color of red found in our capillaries and in our glomerulus is showing the osmolarity. So notice that at the top here in the cortex of our kidney, all of the tubes carry blood that is just as salty or just has the same osmolarity as your blood in the rest of your body. But that countercurrent multiplication, the fact that the descending tubule is right next to the ascending tubule of our nephron loop, helps it so that we can create these really extreme gradients near the deepest parts of the medulla. And that's really, really helpful because then we can use this gradient to perform the work of reabsorption. So these crisscrossing capillaries that are all over the place are really important not only for the function of the nephron, but also to keep it have enough oxygen, have it keep all of the nutrients and energy that it needs to do this hard work because active transport requires a huge amount of ATP. So this is a very comprehensive diagram from your book that just shows where certain materials are reabsorbed and where certain materials are excreted. Notice that everything is initially filtered, and then reabsorption, things we want to keep, secretion, things we want to get rid of that are in excess or that are no longer needed. And then again, reabsorption through the nephron loop, a lot of ions, and of course water. We will still be secreting urea, which is that byproduct of protein metabolism. And then the distal convoluted tubule, we are going to do a lot of that reabsorption of water. And the rest of our reabsorption or secretion will be variable depending on the state of the body. And that can be driven by hormones, it can be driven by nervous signals, and it can be driven by just the chemical nature of the kidney itself. So antidiuretic hormone is the primary hormone that's really going to determine how much water is reabsorbed in this collecting duct and how much is going to remain in the urine. So we talked about this a bit on Monday, that one of the primary diagnostic tools that's used for your urinary system is urinalysis, which is exactly what it sounds like. We're going to analyze everything about our urine. So it could be its overall volume, how much fluid goes in versus how much is excreted, and also looking at it as far as its chemistry is concerned. We can even look at it under the microscope. Normal urine should not have any cells in it. There should be no bacteria, there should be no white blood cells or red blood cells in that urine. So normal urine you should produce from 1 to 2 liters a day. It should be mostly water with just a little bit of salt and of course that urea and other substances that are excreted, but we should have no protein, no glucose, and then no cells in it. So if we see any of these substances in our urinalysis, then there has to be something wrong with the kidneys that are causing this to happen. Many diseases that alter your metabolism, like diabetes or kidney failure, will change the composition of urine, either in the amount that's produced or the substances that are in it. And of course, the most common reason for urinalysis is to detect drug use. That can be an over-the-counter or prescription medication that they just want to see how effectively your body metabolizes it or how quickly it's cleared from your system to make sure your dosage is correct. Or of course, it can be illegal drugs as well. Most of those toxins have to be excreted from the urine in order to get rid of them. So a very effective, quick tool that can be used in a lot of different ways. So it can be assessed if you have urinary symptoms, but it can also be used to diagnose things like pregnancy or like we were talking about drug use or how effective a certain medication is going to be. So you can see all the different things that they can be looking for. We're going to be doing a urinalysis lab with fake urine next week that will be using some of these test strips to test all these different chemical aspects of urine. So that's finally, we're done with the kidneys. We do still have a couple of our organs that we have to talk about to wrap up our discussion of the urinary system. Just remember that once the urine leaves the collecting duct through that renal papilla and into the minor calyx of the kidney, it is not changed. So that urine simply is being held and transported until it can be excreted from the body. So its next step after it drains through the kidney itself is to go through a muscular tube called the ureter. The ureters do have smooth muscle that line them that create peristalsis. Also hydrostatic pressure, just the buildup of urine in the kidney, will push already existing urine through that ureter, and gravity. Just being upright, even sitting, your kidneys are higher than your bladder, so that helps with the drainage of fluid. Once urine gets to the bladder, it can hold anywhere from 700 to 800 milliliters. So yes, it is normal for someone to go to the bathroom 4 to 5 times a day because you don't want it to get that full. You're going to be painfully full with your bladder if it gets anywhere above that 500 milliliter mark. That's a lot of urine. So when the bladder fills, it's actually going to compress the ureteral openings where the ureter meets the bladder on either side to prevent backflow. So one of the biggest reasons for urinary tract infections, where you do get a bacterial infection either in the bladder or the kidneys or the ureter or the urethra, is from holding your urine. So if you gotta go, go. That's one of the main reasons I will never stop you from going to the bathroom. It is healthy to void your bladder whenever it feels it's necessary. The urinary bladder and the ureter are the only two substances or the only two organs that have this very special type of epithelium that allows it to be very, very stretchy, called transitional epithelium. The other structures of the bladder, we'll look at the picture because it makes a lot more sense. So feel free to pause me and copy down these definitions if you would like, but we're going to just jump right ahead to look at the picture. So here we have the picture of the bladder. We're looking at the female version of this because the male system gets a little more complicated because it's integrated with the reproductive system. So the bladder itself is made up of a muscle called the detrusor muscle. It does have these kind of folds in the back called rugae. That's actually the same name we used for the folds of the stomach because both of these organs have to be able to expand and then contract using the muscles in their walls. You can see that each ureter actually has its own opening called the ureteral openings. They're kind of halfway between the top and the bottom of the urethra, or between the top and the bottom of the bladder rather. So you really should not have filling of the bladder past these openings because it just won't work because of the hydrostatic pressure. When we connect these two ureteral openings with the internal urethral orifice, just that passageway where we go from bladder to urethra, it creates a triangular structure that we call the trigon. And this is actually where the muscles can do the strongest type of contraction. When you empty your bladder, there are some smooth muscles that work, like this internal urethral orifice. It's a sphincter, or there's a sphincter around it. The orifice is kind of the opening, and the sphincter is that circular muscle like we saw in the anus, in our digestive system. And just like our rectum, the internal sphincter is involuntary. So when it opens, when it dilates, that is when you feel like you have to go to the bathroom. And then the actual physical control of urination is because of the external urethral sphincter, which that actual opening being the orifice. So an orifice is an opening, a sphincter is a muscle. So the actual process of urination clinically is called micturition. And as I mentioned, it begins as an involuntary muscle contraction, but then hopefully, since you're about 2 or 3 or 4 years old, you can control that reflex. That will create the voiding of the bladder, and the urine will pass through the small tube called the urethra to the external part of the body. So we already mentioned this earlier in our notes for chapter 26, but in males, the urethra is very different. It's much longer, and it passes through an additional organ called the penis. And it also is the creation of the merging of the vas deferens and the urethra in the prostate gland. So that first initial section of the urethra is called the prostatic urethra because it's going through the prostate gland. From there, it passes through a membrane called the perineurium, and it becomes the intermediate urethra. It's very, very short, only a few millimeters. And the longest and most distal part of the urethra is called the spongy urethra. It's named because of the spongy erectile tissue that will surround it. And so the point of that spongy tissue we'll talk more about in the reproductive system is to keep that urethra open, especially for sexual purposes. So when the penis is erect, it does not kind of close up that urethra, but also comes in handy for the release of urine as well. That spongy tissue just helps keep that pathway open. So we will revisit this view of the male urogenital system, of course, in Unit 10. So you can see here's our bladder, and if we trace it down, this is going to be our prostatic urethra, and this is the seminal vesicle. So this little gland here is what creates most of the volume of the semen, and so it does turn into the intermediate urethra, and then it's spongy urethra the rest of the way out. One of the grosser facts about the urinary system that I really like is that most of the amniotic fluid that helps to surround and protect a baby in utero is actually the baby's urine. And the baby will swallow that fluid and then pee it out again continuously. So it's actually a really important part of the development of the muscles of the pharynx that are allowing for swallowing, and for the development of the urinary system itself, because that swallowing and excretion of that amniotic fluid is really, really important. That amniotic fluid is also used for practice breathing to strengthen the diaphragm before birth. The lungs are not inflated yet, but just by drawing some of that in and then breathing it back out, It helps to strengthen that muscle for when baby is breathing air after it is born. Like most other